mutation are of three types: gene mutation, chromosomal aberrations, and genomatic mutations. When we talk about the gene mutation, so here, because of the nucleotide sequence, if anything gets changed, like anything is getting added or anything is being subtracted from that, when that is happening to the nucleotide sequence, then we call it as the gene mutation, meaning it is at the very genic level. Okay. Now, gene mutation may occur due to change in a single base pair of DNA, and we call that as point mutation. When the mutation is occurring because one pair or a single base pair is getting affected or changed, then that is what we call as point mutation. Then comes frame shift mutation. Now, do you know about all of these nitrogenous bases? Adenine, guanine.
Yes, ma'am.
Yeah, you know? So these are the nitrogenous bases, right? And always remember that adenine pairs up with thymine. Guanine pairs up with cytosine, right? This is the pairing. Now what happens is that adenine, guanine, they come under a class of nitrogenous base which we call as purines. Whereas thymine and cytosine, they come under the class which we call as pyrimidines. So purines and pyrimidines are there. Okay, so now let us see. See, frame shift mutation. First, I'll tell you about that. What happens is that whenever the genetic code is made, okay, it is read like it is normally like A, T, G, C. It can be random things like that. Okay, random meaning all the nitrogenous bases, they will be arranged in a sequence, and the sequence can be very random like this. For example, if this is the sequence, okay. Now, in our body, whenever any protein has to be expressed or anything of that sort has to happen, this codon is always read. Okay, reading of this codon is done, and reading is done in the form of triplet, meaning 3 of the base pairs will be read together. So this 3, this 3, this 3, and so on. Okay, so 3 in triplets only the reading will be done. Now, frame shift mutation can be of two types: deletion or insertion. Now, if I remove this C from here, then the whole entire reading frame will be changed, isn't it? Because now ATG will be there, but now GAT will be there, isn't it? It's shifted. So this kind of mutation, either because of the deletion or because of the insertion of any nitrogenous base, it changes the frame that was being read. So we call that mutation as frame shift mutation. Got it?
Yes, ma'am.
Now see, the next type of mutation is substitution mutation. So substitution, just like how teachers are there, substitute teachers, isn't it? When the original teacher is absent, substitute teachers are there, isn't it? They teach very bad, but they are, right? So substitution mutation, it is just like that, meaning now the base pair which was originally there, it will get substituted by another base pair.
Yes.
Now, if a purine is being substituted by another purine, let's just say if there is a biology teacher, okay, and now the biology teacher is absent, I am absent. But in my place, another teacher is coming, but she or he is also a bio. So this kind of mutation will be transition. It just transits, right? Meaning, I'm purine, another person who's coming, that is also purine. Yes. So this kind of mutation wherein a purine is getting substituted by another purine, or a pyrimidine is getting substituted by another pyrimidine. So that is what we call as transition mutation. Okay?
Yes.
Then we have transversion. Now, I'm the bio teacher, maybe I am absent. So in my place, maths teacher, physics teacher, any other teacher came to teach you. So that kind is here when a purine is either getting substituted by the pyrimidine or the pyrimidine is getting substituted by the purine. So that is what we call as transversion. So substitution mutation is of two types, transition and transversion. Clear?
Yes, ma'am.
Now, gene mutations, they are spontaneous. Spontaneous, you understand?
Continuous.
They
are continuous?
No. Spontaneous Continuous means that they are happening like lagatar. That is continuous.
Yes.
Spontaneous is achanak se they happen.
Okay.
No one knows the reason why. Where does
Oh,
it come from? No one knows. It just happens spontaneously, right? How we make plans spontaneously, just like
yeah.
that. So gene mutations, they are spontaneous mutations. They occur due to certain internal reasons that we don't even know. How can we see the genes and everything? This much is
Oh,
clear.
yes, ma'am.
Now coming on to this one, mutagens. Now all those agents that cause mutation, they are mutagens. Okay, mutation causing agents are called as mutagens. They are of
Oh,
two types, physical mutagen and chemical mutagen. Okay, so maybe
okay.
because of certain chemicals like base analogs, acridines, these are some names of chemicals. So if a person is exposed to these chemicals, then the person can have mutation in its body.
Okay.
Physical mutations are those mutations like radiations, UV
Okay.
rays, okay,
Yes.
X-rays, right? So
Yeah.
that is why, if you may know, have you ever had X-ray done?
Yes, ma'am.
So if you remember, the person who operates the machine does not go in with you. It's only
Yes.
you that can go inside, right?
Yes.
And the person who is going, he wears a vest or a
Yes.
protective gear, right?
Yes.
If you remember.
Yes.
Why? Because the
They are.
X-rays, they are very harmful rays. Very. Okay, it's like because of extreme importance, only the person undergoes that test. So X-rays can penetrate in the skin. It can go up till the DNA level and cause
Yes.
mutation because X-ray is a mutagen. That is
Okay.
why the person is wearing protective gear. Clear?
Okay. Yes.
Now, there is this question which I want you to make. See, the question says, no, you make it, then I'll explain. Wait, 1 second. The answer is written here only. I have to rub it. This is related to the monohybrid cross. Okay. So try to solve this question. I hope you can do it. Okay. Can you see the screen?
Yes, ma'am. Mhm. Ma'am, A is blue.
Yes, done, dear.
1 minute.
You're doing the C part.
Yes, ma'am. Done, ma'am.
Tell me.
B is one white flower.
B.
Yes.
They're asking the percentage.
Percentage.
Okay, okay. Tell me C.
C is 9:3:3:1.
Okay, how? Tell me how.
We have to do dihybrid cross, right?
Why dihybrid? That's
The-
that's why you took so long. How did you
yeah.
make this dihybrid? See, only
So
one character is there. Flower color. That's it.
no,
It's a
for the
monohybrid cross. Huh?
for C, I'm saying.
Yes, for C only.
So only selfing is needed. Okay, so the ratio will be 1:2:1.
For genotype or phenotype?
Phenotype.
Then
Genotype.
for the genotype.
Genotype it is B, capital B, small b, capital B, small b, small b, small b.
Those are the gametes that you got for the F2 generation, isn't it?
Yes.
Okay. What is the ratio of the phenotype?
1:2:1.
What is the ratio of the genotype? What is the genotype?
Oh, it's confusing.
First, tell me what is genotype.
Genotype is the ratio.
Genotype, phenotype, both of them can be made in ratio. But my question is, what is a genotype? Like, what is the difference between genotype and phenotype? If you don't know the terms, how will you
Phenotype,
make the question?
phenotype is the BB and
No. Phenotype is what the B and small b represents. So
okay.
phenotype is the external character. Okay.
Yes. Okay.
Genotype is the genetic makeup, like capital B, small b, small
Okay.
b, small b. Okay. So now tell me what is the ratio of the phenotype?
Phenotype is 1:2:1.
Again, phenotype, phenotype. Phenotype is external appearance. It doesn't matter whatever the genotype is like. If I say capital B is tall, capital B small b is also tall.
Yes. So it will be blue, 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 and white.
Yeah, so what is the ratio?
3:1.
Yes. The phenotypic ratio is 3:1. The genotypic ratio, you are correct, 1:2:1. Okay? The first answer is also right. The third answer, you did a mistake because you didn't know the difference between genotype and phenotype, right? That is why I said the basic terms are very important in this chapter. Now coming on to the second part. See, after you made the whole entire selfing and everything, these are the offsprings that you received.
Yes.
Right?
Yes.
How did you make it a dihybrid cross? Like, what did you do to make it like you crossed them all with the same thing?
Yes.
Okay, got it. Okay, but this is always pay attention to what the question is asking. See, they only mentioned color of the flower. Is there any other character? No, right?
No.
They said
No.
blue color flower and white color flower.
No.
Both of them
Yes.
are colors. So always pay attention to what the question is asking. So this is
Yes.
what you got in F2 generation, isn't it?
Yes.
So total you got 4 offsprings in the F2 generation. Now whenever any kind of percentage is asked from here, so they ask the percentage of the white one, right? So how many of them are white? Only one of them is white. Upon the total number of offspring, so we have 4 into 100. So
Okay.
how much are we going to get? We're going to get 25.
Okay.
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 And if you think about it, if not like this, but also you can do it like one more way. Here you can divide 100 into 4 quadrants. So you have 25% this, 25, 25, 25. All in all, you get 100, right?
Yes, ma'am.
Now this much is clear.
Yes, ma'am.
Now tell me one more thing. If I ask you what is the percentage of heterozygous blue?
Heterozygous blue. 75%.
How?
There are 3 blue.
What was my question?
Heterozygous.
What is the meaning of heterozygous?
Okay, 125%.
Heterozygous. Look
Mm.
carefully.
Okay, only 25%. Only one.
Which one is heterozygous?
B. Capital B,
Which?
capital.
This is why I tell you to remember the terms first. Hetero means different.
Okay.
This and this, these are heterozygous blue.
Okay.
So the percentage is 2/4 into 100. We have 50% of them is heterozygous blue. Heterozygous means capital B, capital T, small t together. And homozygous means when both the alleles are same. This and this are homozygous. One is homozygous dominant, one is homozygous recessive. Is it clear?
Yes, ma'am.
Sure?
Yes, ma'am.
Okay, so these are the kinds of questions that may come, may not come also. Now, coming back to the mutagens. So the mutagens, when I said physical mutagens, these are the radiations. Okay, this is the most important type of, you can say important as in this is more common. So we have ionizing radiations and non-ionizing radiation. Now the ionizing radiations are much more harmful as compared to non-ionizing one. So X-ray, gamma ray, alpha ray, beta rays, all these rays are ionizing radiation, whereas UV rays are non-ionizing radiation. Talking about the chemical mutagens, so they are much more injurious than the physical mutagen, such as nitrous acid, acridines, base analogs. These are the names of certain chemicals which have the potential to cause mutation. Clear?
Yes, ma'am.
Any doubts up until now? Okay. Okay, you note down till here, then we'll move forward.
Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am.
Okay, any doubts up until now?
No, ma'am.
All right. Now coming on to the next topic, that is polygenic inheritance. Poly means many. So when one type of inheritance is controlled by more than two types of alleles or more than two types of genes, So then that is what we call as polygenic inheritance. The best example here is human skin color. Now, if you see that there are many people in the world that have extremely dark skin, right? Extremely black, completely black. But then there are again different types of humans also that have completely white, pale skin, right? And then there are so many in between, darker than that, lighter than that. So the inheritance of human skin color is polygenic inheritance. It is controlled by three genes or three traits: capital A, capital B, and capital C. Okay. So like the person, the human that has capital A, capital B, meaning all the dominant allele, they would have the darkest skin tone. And the ones that will have this like the most recessive alleles, so they'll get the lightest skin tone. But then again, there can be other organisms, humans, with this type also, isn't it? There can be this type also, isn't it? There are so many possible outcomes from this, and hence the variety of skin tones that are there in the whole entire world. So the phenomena when traits are controlled by three or more than three genes is what we call as polygenic inheritance. And remember, the effect of each of the trait is additive. Additive means it is like a collective effect of all of the genes or all of the allele that are present in the genotype. Every allele will present some sort of character, right? Now, if you might see, there are some people, like, do you know undertones? Skin's undertone?
Yellow,
Yeah, gray. Yes, very good. So, like, different people have different undertones also, right? Some people have a yellow undertone, some people have a pink undertone, a gray undertone, right? Neutral undertone. So, depending upon that, that also varies from human to human, right? So, that is the reason why they say that effect of each allele is additive in nature. It gets added up, and each allele contributes to the traits of the phenotype, such as human height, human skin color, then kernel color of the wheat seed color. That is also a result of polygenic inheritance. Clear?
Yes, ma'am.
Now comes pleiotropy. Now, pleiotropy is that phenomena when one gene that is present in the organism shows multiple effects of the phenotype. Then we call it as pleiotropy. One gene, many effects. Okay, this is called as a pleiotropic gene. The best example here is a disease which is called as phenylketonuria. Okay, now here this disease is actually caused because of mutation in the gene. So normally in our body, there is an enzyme called as phenylalanine hydroxylase. This enzyme is produced normally. But if the gene for this enzyme gets mutated, now this enzyme will not be formed enough. So occurrence of this disease is expressed phenotypically by mental retardation, reduction in hair, skin pigmentation. So see, because of the absence of that enzyme, or you can say because of that mutation, so many phenotypic effects are seen in the person. Like the person will be mentally retarded, they'll have reduction in the hair and also a lot of pigmentation on their skin. So one gene that got mutated, because of it so many effects are seen. So this is the best case, you can say, example of pleiotropy. One gene, many effects. Clear?
Yes, ma'am.
One more example in the case of plant is starch synthesis in the pea plant. Okay, this is also one example of pleiotropy. Any doubts?
No, ma'am.
All right, now we have Mendelian disorders. Now, Mendelian disorders and chromosomal disorders, these are the two types of disorders that you have to study in this chapter. Mendelian disorders are those in which mutation is happening in the single gene. Okay, so this is like all the examples of these diseases or disorders. It is occurring because there is a mutation happening in single gene. Such as, now the examples of all of the Mendelian disorders, we have hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia. Okay, these are all the disorders. Now, Mendelian disorders, they have two types of natures: sex-linked recessive, sex-linked dominant. Okay, Sex linked means it is related or it is connected with the sex chromosomes of the human. Okay, and if it is autosomal disease, then that is related with the body chromosomes of the human beings and not the reproductive cells. So this is like a nutshell representation of what kind of disease that is. So for example, if we talk about hemophilia, so it is a recessive disease. Okay, meaning, yes, please answer this correctly. If it is a recessive disease, in which condition will it be able to express itself?
In genotypic?
Yeah, in genotypic, like which genotypic condition will the recessive disease express itself?
Genotypic recessive.
What is that condition called? Recessive is correct. Before that, एक और word है. What is that word?
Heterozygous?
Think before you speak. All in all, how many conditions are there?
Homozygous and heterozygous.
What is
Homozygous?
homozygous what?
Recessive.
Yes. Always remember, homozygous recessive means they will only express themselves when they are homozygous in condition, right? So X-linked recessive disease means whenever it will be in a recessive homozygous condition, then only this disease will be shown in the human being. Color blindness. Color blindness is also X-linked recessive disease. Then we have sickle cell anemia. So it is autosomal recessive disease. Autosomal means body related to the body cells and not the germ cells. Then all the other diseases, they are autosomal diseases only and recessive kind except for myotonic dystrophy. Myotonic dystrophy disease is autosomal dominant. Rest all of them are recessive in nature and most of them are autosomal except for hemophilia and color. Clear?
Yes, ma'am.
Now we will be discussing each of these diseases one by one. So the first one is color blindness. Now see, the color blindness is X-linked, right? X chromosome is there in females also, in males also, right? Now the X chromosome, now normally the X chromosome is there in the body of both the organisms, but the gene that will be carrying the color blindness will be XC, meaning it is carrier of color blindness gene. Okay. Now, if this is the case, XC, Y, okay, meaning the carrier X with the Y, then this is affected male.
Okay.
Okay. XCXC, that is affected female. But here XCX is the carrier female. Now tell me, this is condition number 2, this is condition number 3. Now tell me, why is the third one a carrier and the second one affected? What is the difference that makes them so different? See, affected means bimari hai. Carrier means they are carrying the disease, like gene for that bimari, but bimari hai nahi.
Okay.
Okay, so tell me, why is this happening?
Because it happens only in females.
Why? It's happening in male also, the first one.
Yeah.
My question is, third wale mein, why is it a carrier? Why not affected?
Because it doesn't have C.
So answer me in biology.
Because it doesn't have C carrier, C, XC.
Okay, it doesn't have XC. So what does that mean?
That there is no linked recessive.
Think and answer again. Question समझ में आ गया ना?
जी.
The answer is very easy. अभी बताया था तुमने मुझे, थोड़ी देर पहले ही बताया. तुमने नहीं बताया.
This is so confusing.
It is not. It is, I'll tell you, the only reason it is confusing is because you haven't learned the terms yet.
Yes.
See, always remember which kind of disease is color blindness. I've written it here.
Because it is recessive.
Which kind of recessive?
Homozygous recessive.
X-linked recessive disease, isn't it?
Yeah.
It
Yes.
means that when it will be present in a homozygous condition, then only it will get expressed.
Yes.
Right? So that is
Yes.
why the second one is affected, but the third one is a carrier because here it's
Because-
not present in homozygous condition, it is
yes.
heterozygous. So this is
Yes.
not getting affected. Carrier is there, but not getting affected. Tell me about the first one.
It
This
is
is not,
also
this is
zygous.
not recessive, right? Homozygous recessive is not there, but
Yes.
why the male is affected?
Because the male has XY chromosome.
That's true. So
It has to be heterozygous.
to explain it, you are very near it. You're like circling around the answer. You're very near it. Tell me, why is the male getting affected in spite of having homozygous recessive condition? In spite of not having homozygous recessive condition.
Because it is X-linked, so
So
the X is recessive here.
X is X. Normally, if you think about it, color blindness is a disease that is X-linked recessive disease, isn't it? Meaning,
Yes.
when the carrier, when the XC, this gene, will be in a homozygous condition, then only it will express itself. This much you've understood, isn't it? My question is, why in the male there is no homozygous recessive condition, but still the male is affected?
Because the male has only one X chromosome.
Yes, because the male carries only one X chromosome. My questions are not complicated. They are very basic questions. Always I tell you this. Because the
Yes.
male does not have two chromosomes. So how will it have a homozygous condition? It will always have this condition. When the normal X and Y will be there, so then the male will not be affected. Right? So if I talk about the affected male, tell me which of them will be affected male.
First one.
What about the third one? Why not this?
Because the it is a X-linked disease, X-linked recessive disease.
Very good. Clear?
Yes, ma'am.
All right. Now moving forward, so basically here when we talk about this disease, so we see that because of the X chromosome, the gene gets inherited to the person, right? Now if you think about it, the male also gets this disease, right? Whenever the male gets it in a heterozygous condition, and the X-linked recessive diseases, they have a criss-cross inheritance, meaning a male will be transmitting his trait to his grandson through the daughter, and a female would transmit it to her granddaughter through her son. Okay, but this is what is meant by criss-cross inheritance. Yeah.
Okay. Yes, ma'am.
Now, see, basically in color blindness, what happens that a person who has color blindness, have you ever heard about it?
No, ma'am.
See, a person that has color blindness, it's not that that person doesn't see any color. The person cannot differentiate between certain colors, such as blue, red, green. They can't see the red and green color properly. They can't differentiate in between them. So that is what happens in them because of this particular disease. So color blindness is a sex-linked recessive trait in which the affected person fails to distinguish the red and green color. Now, how is the this thing disease inherited? See, whenever a normal woman XX will be marrying a this thing, colorblind man. So if you see the inheritance, it would be that out of the 4 offsprings that they get, 2 of them will be the carrier of diseases and 2 of them will be normal. Okay, how? XCX, that is a carrier female. XCX, that is again a carrier female. But XY is unaffected male. XY is unaffected male. So whenever a normal woman marries a colorblind man, so 2 of her children will be carriers and 2 of her children will be normal. Okay.
Yes.
The second condition is when a carrier for this woman. Okay, you only tell me. A carrier woman marries a normal man. Tell me what will be the genotypes?
The same.
The same.
Do I have to do it?
Do it. Maybe you are right, maybe not.
Okay. Yes, ma'am. 2 children are normal.
Hmm.
2 will have the color blindness.
2 will have the color blindness or something else. Are these the offsprings that you're getting?
Yes, ma'am.
Okay. Then how 2 of them have color blindness?
XCX and XCY.
XCX is a colorblind woman or female.
Yes.
Which kind of disease are we talking about?
Color blindness.
Color blindness, right? Right now,
Yes.
3 times you have told me the right answer. Still you are confused. Is there a difference in between them, Naman?
Okay,
Is there a
so
difference?
female will have XC and XC.
Which one?
The first one, right?
See, I've given you 2 conditions, right? XCX, XCXC.
Yes.
Are these the same thing?
No.
What is the first thing?
Color blindness in female.
And second?
Color blindness in female.
When I gave you the question, I said a carrier for color blindness female is marrying a normal man.
Yes.
And the carrier for colorblind female is this. So how
First,
is this? She will also be a carrier only, na? Color blindness is expressed in homozygous condition, no?
so the second one.
No,
Okay.
see, the gametes, the offsprings, the genotype that you have made, it's absolutely correct. This crossing is absolutely correct. Now I'm asking about the phenotype.
Okay, phenotype.
Now you understand what I'm trying to say. You said that 2 of the children will be colorblind and 2 of
Yes.
them will be normal. My question is, look at it again and tell me how many of them will be colorblind.
Two.
2 of them will be colorblind.
Yes.
Okay. Tell me the genotypes of those that will be colorblind.
XC, XC. and XCXY.
XC. Where are
Yes.
you getting that from?
No, XCX
Hmm. So
and
XCX is not a colorblind woman. She is a carrier for that disease.
okay.
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 Why? Because the colorblindness have to be in this condition or a homozygous condition. Then only it will be expressed. Or else, like the female will be carrying it. She might give it to the offspring, but she is not colorblind.
Okay.
Okay. Now make a cross of this one. A carrier woman with a colorblind man. A carrier woman with a colorblind man.
Mhm. 2 will be the carrier.
Yes, tell me.
2 are the carriers. And
You
2
were crossing what? The carrier woman with a affected male. Tell me the genotype of the parents. What will be the genotype of carrier female?
XCXC, XCY, XCX, and XCY.
XC,
Y, XC,
Y.
and XCY.
XCY is there. XC, then
XCY, XCX, and XCY.
So how many of them are like we are crossing out the carrier female with the affected male, right? So we'll have XC, isn't it? Then we'll have XCY. Then we'll have XCX and XY, right? So tell me how many of them are carriers and affected?
2, two
Tell
are,
me. Huh. Two of
and
them
2,
are
and 1 is carrier, and 1 is
and
not.
one is unaffected. Understood?
Yes.
Clear है ना पक्का एकदम? Good, very good. All right, so color blindness is clear to you, right? Now we're moving on to the next, that is hemophilia. This is also the same kind, X-linked recessive, okay? But here the disease is different. In hemophilia, defective blood clotting factor is produced. Now in our body, blood clotting happens whenever we get injured, but in the people that have hemophilia disorder, in their body, blood clotting factor is faulty. Faulty means not good, defective. So because of which the blood of the affected individual would fail to coagulate. So if this person ever gets a cut or ever undergoes any accident, they'll die from bleeding out. Okay. So basically here the gene is XH, H for hemophilia. Okay. Now the same cases affect here XHY will be the affected male because again this is a X-linked recessive disorder. XHX will be a carrier female and the XHXH will be the affected female.
Okay.
Okay, it will be affected only in the homozygous recessive condition.
Okay.
Now, the possibility of a female becoming hemophilic is very rare because in this case, the mother has to be a carrier and the father should be hemophilic, isn't it? Then only we saw that one female was getting affected in color blindness also. So here, if a male is hemophilic, then they can't even survive.
Okay.
They'll die. That is why the female can't be hemophilic. Like there's a very little possibility. It's very rare for a female to be hemophilic. Carrier, she can be a carrier for that disease. But if she is hemophilic, then that means the parent father should be hemophilic, but they'll not survive. They'll die. Also, the hemophilic female also dies before birth.
Okay.
So normal man, and then a carrier for hemophilia. Then if you cross this, you'll get all those genotypes. Only thing to remember over here is that whenever the X-linked disease has to express itself, it will express itself in the homozygous recessive condition and not in any other condition. Clear?
Okay.
Now, the third disease is sickle cell anemia. Now, sickle cell anemia is that kind of a disease which is autosomal recessive disease. Meaning now it's not linked with the X or Y chromosome. It's linked with the autosomal, meaning body chromosomes. So sickle cell anemia, the gene which carries this is known as HB. Okay, that is the name of the gene, and the one that is carrying it S. So if HBA homozygous condition is there, then that means the normal homozygous individual is there. Okay, it doesn't tell whether it is a male or a female because this is not X-linked disorder.
Yes.
HBA and HBS, which is like this thing, heterozygous condition. So here also the individual will be normal, but they will be a carrier. Since this is also recessive disease, autosomal recessive, so they'll also express in homozygous form. So this one, HBS is the homozygous form, which is the mutated gene. So here the individual will be diseased and they die before attaining maturity, meaning they don't survive for a very long period of time. Why and what happens in this disease? Basically, this disease is also a result of mutation. Here what happens? It is caused by mutation. Which kind of mutation? Transversion. Transversion. What was transversion?
When the one gene is different from the other, like if pyridine is there, then there, like they are
No,
two
both of them will not be present together.
yes,
So what is happening?
like they are two different.
What did I say? Bio teacher is absent, physics teacher is coming to teach you.
Yeah.
Right, so
So
I am not there.
yes.
So when one gene is substituted by another type, purine is substituted by pyrimidine. Very good, you remember. Okay, so here also transversion happens of the gene controlling beta chain of hemoglobin. See, hemoglobin has alpha chain, beta chain in it. So here the beta chain of the hemoglobin gets affected. Okay, and the hemoglobin that is affected is HBS. Meaning after being affected, it becomes HBS. Normally, it is HB. HB is for hemoglobin. Now, HBS causes one change in the amino acid sequence of the beta chain. Here, the amino acid glutamate, this is very important line. Here, the amino acid glutamate changes, amino acid glutamic acid gets replaced by valine, another type of amino acid, at the 6th position of the beta chain. So imagine it has certain amino acid sequence, but now one amino acid is replaced by another amino acid. A different amino acid is coming. So here, because of which, what happens? This causes polymerization. Like, you know, what is the function of RBC? Do you know?
RBCs, red blood cells. No.
See, RBCs, they are the red blood cells. They contain hemoglobin and it carries gases throughout our body, oxygen and carbon dioxide. Okay. And if Our body wants the hemoglobin to carry more and more oxygen. So that is why RBCs do not have nucleus, so that they can carry more. Okay, normally the RBCs are like this, have space for the oxygen to be present. But because of this mutation, from this shape, the RBC's shape become like this.
Okay.
Sickle-like. Okay. Now tell me, will this RBC carry more oxygen? No. So because of this, the oxygen carrying capacity of the RBC is reduced. So in our body, oxygen will not be supplied properly. So this disease is known as sickle cell anemia. If iron, or if people say that खून की कमी हो गई, that is anemia, right? So sickle cell anemia, the anemia is happening because of change in the RBC's shape. And this is a genetic disease. It is a mutation disease. Clear? Then we have phenylketonuria. Now, phenylketonuria, this is a disease in which metabolism error happens. We already discussed, right? Because here in the pleiotropy, we discussed this, one gene, many effect. So here the enzyme phenylalanine hydroxylase will not be synthesized properly. And what is the function of this enzyme? This enzyme converts the amino acid phenylalanine into another amino acid tyrosine. So if this enzyme is not synthesized, then the amino acid will not be changed. And the accumulation of phenylalanine causes that mental retardation and many effects. Clear? That is why the normal synthesis of this enzyme is very, very important.
Okay.
Any doubts up until now?
No.
Okay. Now we have thalassemia. Thalassemia is yet again autosomal recessive disorder caused by the mutation or deletion. Here, defect in the synthesis of globin chain of RBC takes place. Abnormal hemoglobin is made over here. Okay, abnormal hemoglobin. Now, this is happening because of deletion, right? Something is getting deleted. So, types of thalassemia, there are two types, alpha thalassemia and beta thalassemia. Here, alpha chain is affected, and in the beta thalassemia, beta globin chain is affected, meaning hemoglobin has alpha chain also, beta chain also. So in alpha thalassemia, the alpha chain of the hemoglobin is affected, and in beta thalassemia, beta chain is affected. Okay, and they, like, you know that all the proteins, they have certain chromosomes that code for them. So HBA1, HBA2, these are the genes that are present on chromosome number 16. So this mutation happens on chromosome number 16 in our body. And the beta thalassemia, it happens in chromosome number 11. Clear? Now, if you think about it, in thalassemia, also we are having hemoglobin disruption in one way or the other, isn't it? Hemoglobin is not synthesized properly, right?
Yes, ma'am.
Then in the other diseases, like for example, if we talk about sickle cell anemia, in that case also RBCs that have the hemoglobin, that is getting affected. So what is the difference in between them?
That this is by mutation and deletion, and that is caused by mutation and
You are right. You are on the right path.
it is caused by mutation and
Transversion, that is a substitution mutation. And here it is a deletion mutation. Very good. Okay. So this is the difference. Very good. Now, thalassemia is a quantitative problem. Quantitative means less amount, right? Quantity is less. Like quantity of hemoglobin is less. Whereas sickle cell anemia is a qualitative problem. The quality is bad. Okay. This is also different. Any doubts in Mendelian disorders?
No, ma'am.
Okay. Then we have the genomatic mutation. I'll not teach this today. You note down till here because it will be a lot. All the diseases you've understood? Sure. Okay. These are known as Mendelian disorders. Why? because they are following the rule that Mendel told us about, right? The inheritance, homozygous, heterozygous, it is following that. That is why we are calling it as Mendelian disorders. How are you liking this chapter?
It is a bit difficult for me,
Difficult.
but yeah,
But I hope it's not boring.
it's not boring.
Difficult because you are actually like you have already studied this, but you're paying attention for the first time, isn't it?
Yes.
Like properly understanding things. I'm not bad mouthing you, like
No.
properly understand.
Yes.
Okay, okay. Now you can note down.
Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Done,
Done,
ma'am.
Memona?
Yes, ma'am.
All right. Okay, let's